طيب السلام عليكم شباب. بس مراجعة للجريد 11 Advance البارت 1 يعني. We will upload this for to LMS. First of all, if you just gonna to the end of term coverage, oh sorry, for this one, for the topics, yes, the topics here. If you go to this one, يا شباب, you will find that page 75, 74, and 75. This is the shape, figure 2 and figure 1. Okay, let us start with PowerPoint in DNA technology. The first one talk about شباب genetic engineering. Genetic engineering, which is mean that is a way of manipulating DNA of organism by inserting extra DNA from other organism. We call this guys exogenous DNA. Exogenous DNA. يعني manipulating DNA, we bring it from other organism desired, and we put it in DNA for other organism. This pieces of DNA from other organism, we call it exogenous DNA. And they in the end of term coverage focus on this, which is example of genetic engineering, by the way. So one example of genetic engineering is using GFP protein. Guys, this is protein, fluorescent protein, give a glue for organism. Glue. At what time give a glue? When you focus UV light on this. So jellyfish, if you focus UV light, you will find some have green fluorescent protein. They give light, green light. So what the scientists do, they isolate gene and put it for other organism, insert it to other organism, and this organism under UV light start glue. So GF protein, Shabab, we will study this. I put this in LMS, by the way. This is an example of genetic engineering, and we isolate this gene GFP, green fluorescent protein, and put it for this organism, so this organism start glue. Okay. Now, Shabab, we will go to the question about genetic engineering, or no, we will start in page. We talk about restriction enzyme. What is DNA tools? Tools, Shabab, this is figure. They focus on this figure, by the way, in the book. Tools for DNA. حتى نعمل in genetic engineering, guys, we have tools. There is tools. The first tool is restriction enzyme. Most of the enzyme, guys, is a protein. So restriction enzyme protein recognize and bind to specific places in DNA, then cleavage, cut it yani. Okay, this is very important, Shabab. And restriction, there is endonucleases, which is restriction enzyme, cut viral DNA into fragments. Cut viral DNA into a fragment. Okay, endonucleases. So this is very important. So two type of restriction enzyme, by the way, guys. This is the idea of restriction enzyme: bind and cut viral DNA into fragment. شباب, هنا there is two type of restriction enzyme. One is EcoR1. EcoR1, they make sticky end. They cut from specific place in DNA. We call it GATs. They produce what, شباب, هون? We produce this area, GATs. We produce يعني we cut it from this and make sticky end. لكن the other one is blunt. Another restriction enzyme is a blunt end, which cut straightly like this. Okay, this is a blunt end. طيب, let us answer this question, شباب. It's very important. What is genetic engineering? Manipulating. How can genetic to produce plant and animal? This is the idea from genetic engineering. What is the human genome is very important. Total DNA in the nucleus. How we can study with organism genome by استخدامing tools. One of these tools, guys, we call it what? Restriction enzyme. And the others is what? Is gel electrophoresis. Look at these guys we cut here. Let us go to the gel electrophoresis. This is the third figure, I think, focused in the focus in the end of term coverage material. First شباب gel electrophoresis انتبهوا علي. Gel electrophoresis is one of the tools to make genetic engineering. The most important things to know, guys, إنه they move. This is gel electrophoresis is puts agar and the area for DNA fragment, DNA. Then start gel electrophoresis, separate DNA according into fragments according to their size. Okay, so this separate DNA fragment according to the size fragment by electric current. Stop. The DNA fragment move from negative to positive, from negative to positive. You should know, guys, that smaller fragment move faster. يعني the faster one is smaller fragment. And also this technique used in DNA typing, guys, which is in human genome. Okay, so this is the real photo in the lab. We use this, we put the DNA here, start the current, then under X-ray we will see that the fastest one is the smallest one, moving from negative to positive. What's type of question? Let us go, guys, on the question. We will focus on the question. Uh, uh, no, not this one. Exam coverage, not also this. Ah, this one, Shabab, yeah. The first question is about restriction enzyme. Before, they ask what following process can be identified, both biotechnology and genetic engineering. شو الidea يا شباب? Modification of organism to express a desired trait. شو؟ Both البيوتكنولوجي والgenetic engineering. The idea يا شباب انه manipulating organism, manipulating DNA in order to take desired trait. This is the idea. To take ishi مرغوب, desired trait. So this is a question, guys, which is a high level question. I have the normal question I will show you, but this is for you if you understand this. So this two figure X and Y fragment. So they ask which one is true regarding true. They ask about a true there. So the first one, enzyme one, is the restriction enzyme. This is true, guys. This is the restriction enzyme because he cut. But fragment X is a blunt. Is this true? No, not true. This is not true. So the other one is blunt end, and this is enzyme fragment X. This is sticky, and X is sticky. And yeah, enzyme one is the restriction enzyme, and fragment X is sticky. Yes, this is sticky, which is true. Enzyme 2 is ligase. No, enzyme 2 is DNA ligase. No. So the answer is B, restriction enzyme, and make sticky end, which is eco R1 نشبه. Eco R1. So look at this question, guys. They will bring, for example, for gel electrophoresis, this figure. This is father, this mother, child. They ask you guys about which label, which true, correct, focus on this, correct about X and Y. Shabab, X is negative charge and bigger in size than Y. Yes, Y is small, X is bigger. Why it's we call this guys negative? Because it's close. This one is close to negative charge. X is positive charge and bigger in size B. And X is positive charge. No, X is not positive charge and smaller in size in Y. Okay. So the question they ask about who is the biological sibling for this? father and mother. Look at this, guys. Who is biological sibling? Biological sibling, guys, this band, child one, is correct with father. Okay, this band correct with mother, the same. So child one is is child for father and mother. The other one, this one, yes, they connect with mother. This one connect with father. So one and two. Look at. Let us look at the three. Child three. No, no. This one, no, no. They're not. This fragment not combined with mother and father. So one and two is what Shabab, what is biological sibling. Okay, this is very good question, guys. They ask about yes. They ask about what is if you cut DNA in the fragment, which following enzyme can be cut. طبعا hold the video is the restriction enzyme. Let us go. طيب يا شباب, if we put it in the if we put it in the gel electrophoresis, what is that? It is C A B. Why? Because C is the largest fragment. Largest fragment, so move slowly. So the first one C, then A, then B. B is the faster because it is smaller. So you will put C A B. Okay, guys, let us stop in this part. Let us stop in this part. So in this part, then we will continue about genetic recombination. Part 2. I will upload this in LMS, and please just follow the PowerPoint that I give it to you and the page in Endo of term coverage. Thank you.